Good morning, everyone, and welcome to the October edition of DNA Dialogue. As we gather for this symposium, let us take a moment to reflect on the meaning of place, and in doing so, recognise the traditional custodians of all the countries that we're coming together from today. I'm actually joining you from the land we now call Sydney, the country of the Gadigal people of the Eora Nation. I'd like to extend my respect to the elders of this land and all the lands we work and live on, and extend that respect to Aboriginal and Torres Strait Islander peoples joining us today. So, as you probably know by now, DNA Dialogue is a forum for members of the genomics community in Australia and beyond to learn of new developments in health genomics and the overseas experience. DNA Dialogue seminars will be held live on the last Thursday of each month, though we'll soon be conducting a survey of all attendees to date to confirm that this appropriate timing and convenient timing for our seminar series next year. If you're active on social media, please do use the hashtag DNA Dialogue, and I'll just pop in the chat in a minute the other sort of tags you can link to for the purposes of today's seminar. So please do ask any questions throughout the talk using the Q&A function, and I will pepper Dean with those at the end of his presentation. So now it's my very great pleasure to welcome Dr. Dean Regier to our seminar series today. So Dean is a senior scientist at BC Canada and associate professor at UBC. Over the past 15 years, his research has examined the economics of precision medicine, focusing on generating real-world evidence for sustainable healthcare. He's principal investigator for the Canadian Network of Learning Healthcare Systems and Cost-Effective Omics Innovation, or CLEO, which is funded by Genome Canada. CLEO is a patient-partnered project created to inform the design of learning healthcare systems that turn genomic knowledge into sustainable healthcare. So the topic of today's talk is a very timely one for Australia, with Medicines Australia embarking upon their review of health technology assessment, born of the sort of Zimmerman report, the Zimmerman-Freelander recommendations. So precision medicine challenges conventional thinking around HTA, and BC Canada proposes a life cycle HTA framework to manage evidence uncertainties based on real world data collected from learning healthcare systems. So Dean, it's a very great pleasure to welcome you today. Thank you for joining us.
Thank you so much. And I'm just going to share my screen.
Over to you.
Perfect. Okay, well, thank you so much for the kind introduction, Tiff, and also thank you to Australian Genomics for the invitation and indeed opportunity to talk at DNA Dialogue, which is just a fantastic initiative. So I've gone through the previous talks, and really it's a privilege to be able to talk today in this forum about life cycle technology assessments and real-world evidence for precision medicine. Also, good morning to those in Australia, and good afternoon from Vancouver, Canada. Also, being in Vancouver, I'd like to acknowledge that Vancouver resides in unceded Coast Salish territory, which is the traditional territories of the Musqueam, Squamish, and Tsleil-Waututh First Nations. I myself am of white European ancestry from a small town called Swift Current, Saskatchewan, which is kind of in the middle of Canada. And I acknowledge my responsibility to the best of my ability in furthering action on reconciliation. And indeed, to that end, today I'll touch on silent genomes, which Dr. Rob Anand of Genome Canada mentioned briefly in his August DNA Dialogue talk. And as he mentioned, silent genomes endeavors to bridge the genomic divide through building something called the Indigenous Background Variant Library to address underrepresentation of Indigenous peoples in our reference libraries. It's really the IBVL in terms of thinking is similar to the Aotearoa Variome, which is led by, as you know, Dr. Phil Wilcox. In terms of silent genomes, together with our First Nations partners, I am thinking through the value and economic dimensions of the IBVL and the application of whole genome sequencing for diagnosing rare disease. The other kind of thing we're going to touch on today in this talk, as mentioned, is CLIO, which is my network supported by Genome Canada that is really looking at thinking through regulatory value and reimbursement conversations and dimensions of precision oncology with respect to the application of large panels and whole genome and transcriptome analysis in advanced cancers. And so we'll talk about both of those projects today. And it's a bit of a tour de force in terms of my talk with respect to the different projects that I'm going to try to weave together. But I really just, like to reinforce that, precision medicine has a background of a challenging evidence generation for health technology assessment. And there are a number of different reasons for this that we'll talk about throughout the conversation. But against this backdrop of an equitable, effective, and cost-effective implementation of omics and other healthcare interventions and technologies, that precision medicine challenge really becomes a problem for timely implementation of innovative technologies, genomic technologies for the benefit of patients and populations. And really, when we talk about these challenges and their influence or result, is that it results in evidentiary uncertainty. And that evidentiary uncertainty complicates this implementation conversation. And we put forward what we hope is a solution to the challenge of uncertainty, evidentiary uncertainty, through something called life cycle health technology assessment. And indeed, the headline concerns.
Sorry to interject. We can't actually see your slides.
You can't see my slides? That's so sad. Hold on.
Were they very beautiful?
Oh, I gotta say, they were like my favorite slides. Hold on. Is that better?
Yes, I can see those. That's in presenting. Beautiful.
So, okay, so here is the research report on silent genomes and CLIO. Actually, I was before talk while we were talking. I was saying, hey, I could talk for hours without slides, just about the ideas. But we'll try to get the slides for use. Anyways, so let's go back to the headline concerns. And the headline concerns in Canada, as well as internationally, really starts with this conversation around the costs of sequencing and the costs of healthcare. And on the top right, we have some work we've been doing alongside the Personalized Oncogenomics program in British Columbia, which implements whole genome and transcriptome analysis in advanced cancers. And we've been working with them since 2012 with respect to mapping out the costs and the consequences. And although folks like Illumina and other sequencing companies have heralded the now, I think a couple of weeks ago, the $100 genome, that is indeed what we see. What I'll say is a myth, particularly against the backdrop of the high cost of analysis. So on the top right, we have the trajectory that shows whole genome and transcriptome analysis has indeed decreased on a per patient basis from $60,000 when we're using WGTA in advanced cancers in 2012 to around $12,000 to $14,000 today. But what we haven't seen decrease is the cost of bioinformatics, so that more in-depth analysis, what we'll call level 2 analysis to more fully characterize genomic landscapes. And there's all the other costs like sample acquisition, like reagents. So even if we have $100 at scale, which I'm deeply skeptical about, there's all these other really important costs that will still contribute to whole genome sequencing and things like transcriptome analysis north of, at least in our case, $10,000 Canadian. The other headline concerns are really related to small benefiting populations, where even if we're able to have signal of benefit in terms of effectiveness and cost effectiveness, there's a lot of uncertainty around those endpoints. And what's driving some of that uncertainty is this idea we're subtyping and subtyping and subtyping diseases, which really drives the parameter uncertainty and complicates decision making, because that translates into what we call evidence for decision making uncertainty. Although that uncertainty, we do see quite a bit of signal. So on the bottom right of my slide here, we have a real world data from BC Women's and Children's Hospital in British Columbia, where we've tracked the diagnostic odyssey of children with undiagnosed rare disease using different approaches. So the blue line is microarrays followed by single gene testing, and the red line is last tier exome sequencing. And here's a Kaplan-Meier that looks at probability of no diagnosis. And what we see quite clearly here in terms of the diagnostic odyssey and trajectory of these children and their families is that the probability of not getting diagnosis is so much higher for clinical tiered CMA testing compared to exome sequencing. And so we do see signal of benefit. And that actually, as you'll see on my next slide, translates into potential cost savings in terms of healthcare. And so while there is signal, you'll see very quickly that the signal is blurred a little bit because of a parameter uncertainty. The other headline concerns that are near and dear to my heart is really the conversation around patient preference and incorporating patient preference into the deliberative decision-making process, where we think through how do patients and families, for example, value the knowledge surrounding cause of a genetic condition, or value the knowledge of having an actual variant or not. We'll get to that just briefly later on in the talk. And then finally, we're going to kind of focus on the problem with on-off decision-making. And by on-off decision-making, I mean, we say yes or no to reimbursed technologies at a specific time and place, and often ignore the underlying evolving evidence base that happens when we trial in research or in other contexts the implementation of our technologies. And if we just say yes or no at one time point, we're ignoring that evolving evidence base, which I think is deeply problematic for sustainable and cost-effective healthcare systems. I think a really good example of evidence uncertainty and the conversation of equity indeed is exemplified in the silent genomes conversation. And so here is a high-level view of an economic model that we've been working on, going on 4 years now, looking at the application of genome sequencing plus that indigenous background variant library versus a standard of care in First Nations children in British Columbia and youth with a suspected genetic condition. And so the context of the talk doesn't let me go in depth about the economic model and the data, the real world data to support the economic model. But what I do want us to take away from this conversation is that when we look at those situations where there is cost savings and quite a bit of benefit to patients and family, it's really driven in our case by the addition of that background variant library. And the reason for this is that the IBVL, the IBVL resolves uncertainty surrounding, for example, variants of unknown or uncertain significance, because the occurrence of VUS is higher because we don't have those background variant libraries to confirm diagnoses. And drives uncertainty. So the idea is that the IBVL resolves some of that uncertainty and can mitigate that diagnostic odyssey quicker than just, for example, compared to even whole genome sequencing alone, and certainly against a standard of care. And so that's really nice to see in terms of addressing equity issues also can address uncertainty around implementation. But of course, it doesn't address the other big problem that I mentioned, which is the uncertainty in the underlying data and data generation processes. And in economics, we represent that uncertainty through these clouds that you can kind of see in the bottom right. And so these clouds, the green is whole genome sequencing plus the indigenous background variant library. So that's green. The blue is research whole genome sequencing. And the plane maps out the incremental costs of those strategies against standard of care and the incremental effectiveness. So cost is on the vertical and effectiveness is on the horizontal. And the takeaway here is that we can clearly see that although we believe that whole genome sequencing plus the IBVL is cost saving, there's quite a bit of uncertainty about the degree of cost savings. So in some cases, it's more costly. In the majority of cases, it's less costly. But also that cloud in terms of how wide it is very wide. And it also points to, well, we're really uncertain, at least from a statistical data generation process, we're uncertain the degree to which it provides, in our case, additional diagnosis. And so that really is meant to kind of be a segue into the need for more data and thoughtful data generation processes that go beyond our current thinking. And so I'm going to talk a little bit today about lifecycle health technology assessment. So this was actually published, this is really new thinking in that it was just published yesterday in MPJ Precision Oncology. And really, it's meant to map out some of our thinking around the use of real-world data, which is broadly thought of as data on patient and health system outcomes routinely collected from a variety of sources, and also how that data informs real-world evidence and our decision-making. And really, whether it's this network that we have called CLIO, or the other kind of initiatives that we're thinking through, it all starts from this concept of learning healthcare systems. And we do know the importance of learning healthcare systems in context to genomics. So on the top left of the screen, I've stolen some ideas from the NHRGI, that the virtual cycle by Green et al. In the Great Nature publication. And also the well-known infinity loop from Genomics England. And both of these kind of initiatives or thought processes or frameworks really coalesce around this idea of, in the case of the virtual cycle, the genomic learning healthcare system, in the case of the infinity loop, evolving genomic healthcare. And all of them are really trying to think through, how do we generate and apply data and evidence to improve patient health in real time? How do we drive discovery as an outgrowth of patient care? And how do we ensure innovation, quality, safety, and value for money in healthcare? Now, as a point of departure in terms of and how an economist like me might think about these conversations, is I deeply believe in learning healthcare, and I do think that they can be sustainable and have both patient and population health impact and indeed improvements. But I do think that we need to think about, and this is what makes us a bit unique, is we need to think about it as an ecosystem where implementation of technology and innovation are both created and destroyed. And by that, I mean, you can see that, what we published in terms of MPG Precision Oncology was one of, we think, life cycle evaluation and learning healthcare system are moved forward through all the types of evidence that we deliberate on, through health technology assessment and reimbursement, through management, through optimal use and monitoring, and to allow innovation and an ecosystem, we need to disinvest, and this is pretty key to the idea, from low value technologies. And why that's so important is that low value technologies have a real opportunity cost to them, not only in terms of the money that we spend if they're not as effective or cost effective as we once thought, but also in terms of making room for new investments. And so if we invest too much in one thing that doesn't give us patient or population health, then part of the opportunity cost is lost health for other patients who don't have the same opportunity to benefit from innovation. So the idea is that we need to invest away from these low value technologies so we can continue to invest to innovation and collect the data that supports ongoing conversations around effectiveness and cost effectiveness. Of course, this idea of learning healthcare systems are not without challenges. Our current, at least in health in Canada and I would guess in many parts of the world, is that our current systems, regulatory and reimbursement platforms, policies, and practices are really preventing what we suggest are developing nimble responsive systems that enable decisions based on discovery and new forms of evidence. And by that, I mean, we talk about a lot about learning healthcare systems, but we are not set up always to realize those learning healthcare systems. And what are those practical steps needed in order to actually have learning healthcare systems that generate data to allow innovation and disinvestment? And so in order to kind of move away from learning healthcare systems and challenges that we all know are associated with learning healthcare systems, this concept of life technology assessment we put forward, as have others, may be a way through generating data for real world evidence and decision making. And as an offer of a definition for life cycle health technology assessment, or life cycle HTA, we figure it's one about standardizing real world data collection, including that data collected in trials, standardizing study designs and methods analyses to guide the appraisal, reappraisal, and the adoption of health technologies. And I really want to be clear about this. It's not about putting out fires and oh, this is lots of uncertainty around effectiveness endpoints and safety endpoints. We should do a real world evidence study. It's really one of starting from the beginning and partnering with omics, with pathologists, with genome scientists, with clinical trialists, et cetera, right at the beginning to ensure that our healthcare systems are generating the right types of data and actually having the right types of study designs to be able to infer some sort of causal estimates of benefit and cost effectiveness. And so we've been doing a lot of thinking around these methods and what's needed to set up life cycle health technology assessment. And I'll go through briefly, just very briefly, some of these methods that we've been publishing on, including study design, which is the one at the top of the screen. In the middle, we talk about a core data set in this case for precision oncology. And today, I don't have enough time to do it justice with respect to how do we engage patients and publics and families with respect to valuing the degree to which, or the extent to which precision medicine is valued. And can we actually trust, and you see trust is a big kind of conversation piece, both in the real world evidence space and in the preference elicitation space, in terms of how do we trust the estimates that our models are producing. So I'll first talk about what we've done in terms of the core data set. So this is published in Valuing Health this past year. And really what drove this idea was, and this is work led by Dr. Samantha Pollard, is really what kind of data do we actually need? And we got a lot of these questions when we engage people. It's like, well, we don't even know what data to collect. And so we said, well, we have a pretty good idea, but is there a more systematic way we can think through in terms of trying to ask questions around what types of data can support health technology assessment, and in this case, cost effectiveness analysis. So we went through a modified Delphi process with international healthcare system stakeholders, including those from Australia, to really map out unique data sets. And we, after this, we came up with 68 unique required data elements that will enable conversations around effectiveness and cost effectiveness. And those data include healthcare resource utilization, quality of life data, preference data, clinical data, genomic data, all carried out throughout the entire timeline or course of an intervention, and also before that intervention. And that's really important when we try to ensure that if we are trying to mimic or trying to replicate potentially a randomized controlled trial in absence of an RCT actually being carried out. We need that baseline characteristic data to look at the trajectory both before the intervention and after, to ensure that some of our analyses are giving us causal information around outcomes. And speaking about causal inference, the study design and analysis is really important for precision medicine. This is with respect to precision oncology, this study design. It's a generalized study design that we've created. And the need for the study design, and this is a real world evidence conversation, is because there is a lack of randomized controlled trials in precision oncology and in precision medicine in general. And that, again, is an outgrowth of small benefiting populations, making it really difficult to conduct randomized controlled trials. Of course, what we know is if we don't have RCTs, causal inference around outcomes, that is incremental effectiveness, safety, and cost effectiveness, is really problematic because causal inference is, to some degree guaranteed in RCTs, but because of bias, selection bias, etc., some of the econometric or statistical techniques that we use to adjust are still problematic in terms of being able to ensure that we minimize or mitigate those biases. And so this is a generalized study design to generate what we call a synthetic counterfactual that is able to mimic a randomized control trial, so we can have some understanding or some conversation around causal estimates around outcomes. And so we've done a lot of this thinking, and we've implemented this thinking in a program called the Personalized Oncogenomics Program. We affectionately call personalized oncogenomics in BC POG. And so these are a whole bunch of POGs in terms of individual patients and visualizations of their genomes. And what POG does is it applies whole genome and transcriptome analysis to characterize genomic landscapes of patients with advanced cancers. And also, when available, use WGTA to guide treatments. And the process is in the circle on the right of my screen, where we go through biopsy, we do WGTA, we also data share, we collect data, etc. And we also report hereditary cancer results, etc. And then somebody like me is trying to think through, well, it's a single arm trial in that it's not randomized, but the intervention is whole genome transcriptome analysis. The question becomes for me, like, can we use our learning healthcare system to generate the counterfactual in terms of what would happen in absence of whole genome and transcriptome analysis, and also to generate additional data around health service resource utilization and length of life of both the intervention and standard of care. And so what we've done, and this has been going on, we've been pursuing this for 5 or so years, is, we've linked data of our healthcare system in British Columbia. We've applied that study design that I just popped up on the previous slide to really answer the question around effectiveness and cost effectiveness about the application of WGTA in advanced cancers in British Columbia. And so this is what we found, and this is published in the Journal of Community Genetics. And so what we found is that on average, and we have Kaplan-Meiers, so the top left is a Kaplan-Meier of whole genome and transcriptome analysis, which is red, and blue is standard care. And on average, we see that those patients, in terms of length of life gains, probability of survival, there's no difference. We move down to the bottom of the other Kaplan-Meier, is that when we tease out those patients who have had POG informed or WGTA informed care, that is, they've had a identified actionable variant and also received treatment based on that finding, and that's the green line. The blue, the red line is patients who've had WGTA but did not have an actual finding and did not receive a treatment because of lack of actual finding, according to that finding. And then the blue is standard of care. And then of course, we've also looked at biopsy failure patients. And what we found through this analysis is that there's signal there in terms of POG informed treatment does improve length of life. And there's a degree of immortal time bias here that we will mention. But there is still a signal when we compare POG informed care versus POG naive care. And indeed, we see that the hazard ratio does infer some survival benefit if you receive POG informed care. Of course, what we also see is that therapy plus genomic whole genome and transcriptome analysis is very expensive, and we summarize how expensive a return on investment through the incremental cost effectiveness ratio, and we found that it's north of $154,000 Canadian, and has a very low probability of being cost effective. We did some scenario analysis where we're like, well, what if we had fewer biopsy failures and a higher WGT-informed rate of treatment? That 45% in scenario 1 is what we're finding today in POG. Well, it's probably between 40 and 43%, to be honest. And we do see that the more patients who are able to have WGT-informed patients, that improves the ICER by quite a bit, but there's still a lot of cost effectiveness uncertainty around what is the right decision in terms of implementation. And then finally, in this scenario 2, we've incorporated through something called a discrete choice experiment, which I will mention, Canada and Australia and to some degree the UK are leaders in the application of discrete choice experiments to outline willingness to pay. And indeed, Australian Genomics' very own AEST Gorantitis does brilliant jobs with his DCEs. And when we incorporate the willingness to pay of patients for information related to whole genome and transcriptome analysis, we have this ICER going down to $75,000 per life year gained, with a bit of a higher probability of cost effectiveness. And so why am I going through that just to get to this final-ish slide, is that even in context to study design, real-world data at a particular point in time, and incorporating patient preferences into that conversation around economic benefit, you'll see that the probability of cost effectiveness is still very low. There's so much uncertainty around whether these interventions, in this case, whole genome transcriptome analysis, although it extends to every aspect of precision medicine that I've been involved with, at least, is still like the decision is still highly uncertain around the value proposition of implementing comprehensive forms of genomic sequencing. And so that kind of gets us to, well, if that is the case, if because of small benefiting populations, because of a lack of randomized controlled trials, really drives parameter uncertainty and then decision uncertainty with respect to implementation, how do we start to address those conversations? And so indeed, that's why we came up with this life cycle health technology assessment, we'll call it framework. And really it's meant to be an iterative decision-making process where, for example, we have a managed access agreement in place, whether that be technology managed access or drug or both. And we say, hey, there is signal there with respect to effectiveness and cost effectiveness, but there's a lot of uncertainty around that signal. So the solution, we think, is collecting real world data and all the data like resource utilization, quality of life, et cetera, as part of everyday care through that learning healthcare system. And then we concatenate that real world data. And this is really key to us with external data sets. I know we talk a lot about in genomics, Cancer Genome Atlas and so many data sharing initiatives, Nomad, et cetera. Genome science is really great at open science and sharing data. And in health economics and health services research, not so much. And indeed, we have rarely concatenate data at the individual patient level if at all. And so we want to collect this real world data and concatenate that data with trial data, with data from other jurisdictions to inform this real world evidence package. And that RWE package looks like clinical effectiveness, cost effectiveness, patient value, and also value of information. And value of information is really important to us because it essentially gives us a quantified estimate of should we continue to collect more data? Have we resolved uncertainties, or more uncertainties, or less uncertainty? And if we were to continually to address the uncertainty around our endpoints, would that be valuable to decision makers? And so the idea is that we have this evidence package on endpoints and outcomes, plus should we continue to trial through a managed access world this new intervention. And of course, we interpret and deliberate, we update our decision, we disinvest if BOI and the other endpoints suggest we shouldn't invest and suggest that's low value, or if it suggests that we need to continue to address uncertainty and there's still a signal around effectiveness and cost effectiveness, then we keep on going through the circle until we get to a point where in a deliberative world, we can really say, hey, this is a low value or this is a high value drug or intervention. And so I think I'm pretty much at time for the things that I wanted to discuss. And my last slide is really just to go over some key points, because I understand, so we started with silent genomes and equity and relative value and cost benefit of rare diseases and the implementation of exomes and genome sequencing. We moved to precision oncology and evidence generation to address the uncertainties. And I guess, the key points, the highlights, if you were to ask me, what you could take away from it is that, precision medicine, whether it's in oncology or rare diseases or in any other space, really faces an evidence generation and equity problem. And we think that, real world evidence from our learning healthcare system using real world data is a partial solution, in that, as we saw through the rare diseases example and silent genomes, that the addition of the IBVL helped us resolve uncertainty, but only to a degree. The rest was data generation problems. And so RWE can help us with that, especially in light of a lack of randomized controlled trials, allowing us for reliable causal inference. But the thing that we still have to grapple with, I think, and I think it's more than a Canada thing, I think it's an Australia thing, I think it's an England and Scotland thing, I think it's a worldwide international problem, is that these components of real world evidence generated from our healthcare system and also patient values through preference elicitation studies using discrete choice experiments, for example, have a ways to go before earning the trust of science. And indeed, earning the trust of decision makers, at least in my experience. And the only way, the best way that I can think about, earning trust is having transparent analyses, enable open science to the degree that it's acceptable, to interrogate data and the veracity of conclusions that have emerged from the real world evidence generation process. And the final thing I'd like to leave you with is that the hope or the thinking around lifecycle health technology assessment is really to flip everything around. I think traditionally, and I'm not always popular when I say this, so you'll have to bear with me, but the traditional way of generating data, at least in the precision medicine space, is to start with discovery and multi-omics, which I think is great. And you have discovery, and then maybe you take it and trial it in a single arm trial, or ideally a randomized controlled trial, and then you ask the economists to come on because we need patient value and cost effectiveness, and then you take it to decision makers. But what lifecycle health technology assessment at its core is really thinking around, that's great and that's really important, but we should think about the ending and equitable access to omic technologies and sustainable technologies, and then move backwards. And so the idea of lifecycle HTA is to use the decision-making process, the implementation process, as a way to generate the data, not to be responsive to the omics, not to be responsive to the clinical trials, but to, in parallel with the omics, with the clinical trials, generate data to mitigate or to address some of the statistical uncertainties that we see in the evidence base. So I'll leave it at that. Thank you for being patient, and I'm happy to discuss the ideas.
Dean, thank you very much. A tour de force indeed. It's certainly a lot of food for thought there, particularly, also in the Australian context with our health technology assessment processes. I was very interested in, I love the idea of the life cycle HTA analysis and having a more sort of iterative process of health technology assessment that incorporates patient and public perspectives and preferences, and it resonates with us. We recently had a MBS, so MSAC, a Medical Services Advisory Committee application for expanded reproductive carrier screening, and it was while the sort of conventional HTA analyses was recognised by MSAC, they really want us to go back and consider more about public preferences, patient preferences, the ethical, legal and social implications that we hadn't captured in our original application for public funding. So I think there is an interesting trend towards consideration of public preferences and patient value in health technology assessment, but it doesn't seem to be confidently applied. Like at this stage in MSAC, it's usually the value of knowing, of a diagnosis.
Yeah. So yeah, thank you in response, and thank you for bringing that up. And I think it goes to one of my parting points in terms of this qualitative and quantitative patient value work that we've been doing for a long time now, 20 years, still has a way to go in terms of establishing itself as part of the science, as part of the deliberative process. Stakeholders, whether it be MSAC or CADIF in Canada, want this dimension of evidence. The problem is that this evidence and how it's generated is not consistently well done from a science perspective. And that's surprising. I mean, we need to do more training, but we have been working on this for so long. It just hasn't filtered to the HTA committees yet. They know they want it, but they don't know exactly what it looks like, right? And so CADIF and in British Columbia would deliberate on a qualitative evidence package for patient value, but that is really haphazardly done. It's not done in an intentional scientific approach. We do it in the literature very well, and there's good examples in Australia being leaders in this. But again, it still seems to be what I call a cottage industry in terms of it's very niche, and it hasn't really filtered out in a way. But again, this is important of kind of hacking the idea of going from omics to implementation, going from that to implementation all the way back, in that if we know we need patient value, then we can start to talk to each other and how to do that in a science-driven approach.
Sarah Norris has a question. Welcome, Sarah. Very well placed to contribute to this conversation. She notes that in Australia, our legislation prevents us from having managed access agreements for technologies and service reimbursed via our medical benefit schedule.
Yep.
Is there any scenarios where a life cycle approach to HTA could work without managed access agreements?
Yeah, that's an excellent question. I had noted that. So the short answer is yes, just take the managed access out and engage in partnership with the relative industry partners. And by that, I mean, in fact, we are implementing this framework right now at BC Cancer with respect to NTRK fusions and entrectinib for advanced cancers. And we haven't actually entered into a managed access agreement. I think it's important to outline, what is acceptable and what we need in order to say yes or no to something. But what we did instead was to say, hey, we want to come up with an evidence generation approach with you. And so the idea was we engaged, in this case, it was Roche, to get a free drug in exchange to essentially trial entrectinib at BC Cancer for those patients with intract-positive fusions. And so, the managed access agreement conceptually can be no managed access and zero cost of drug, but also kind of conceptualize it as a single-arm trial that is meant to, as long as we are trying to address evidence uncertainty through generating new evidence and concatenating that evidence with existing evidence, I think that life cycle approach is really valuable. And I will also note and make a shout out to managed access agreements are controversial in some healthcare systems, and they haven't had the type of impact that we would have thought they would have had, even 5 years ago. So those conversations around the value to the healthcare system of managed access is still one that needs to be solved.
Dean, I have a question. I'm sort of interested. So in molecular profiling, we can stratify people with cancers to such an exquisite degree. And I noticed you've got a sheet of POGs over your shoulder. Is that POGs on the back wall?
I'm on POG management committee, so I get the POGs in the back. Yeah.
Beautiful. So I guess there's a risk in terms of both engaging with policymakers about, there's 120 different blood cancers, and this is the effect of each versus, this is how many people die of blood cancers in Canada every year. So in some areas, we're stratifying patients and people with lived experience of cancer. In some, we're sort of lumping them, for want of a better phrase.
Mm-hmm.
In your work around patients' lived experience and perception of value of interventions, to what extent can that be stratified? Be it from, the type of cancer, can it be from their own cultural background, or those 68 data elements that you referred to,
Yep.
To what extent do they track between patient groups?
So that's a good question that I'll try to answer it in one way, and you can tell me if I'm answering it in a way that directly addresses the idea. And I think blood cancer is so diffuse large B cell lymphoma cell of origin, and I thought it was ABC, DCB. I thought there was like 3 subtypes, and it's exploded since then. And so yeah, we are subtyping cancers. The question is, for somebody like me, is can we tease out or understand patient value in relation to that information? Is there a value related to, for example, for ABC subtype in diffuse large B-cell lymphoma, we know that those patients have a poor prognosis compared to all other subtypes. And so we did some patient and public engagement around this question, really trying to get to that point of, is there value in understanding your prognosis, even if it's a worse prognosis, even if you don't have treatment available? And the broad answer is yes. And the next question is, can we tease out those subpopulations and those other person characteristics, socioeconomic backgrounds, or just individual preference to really tease out and individualize the value of healthcare in context to subtypes healthcare? And the answer is we can do it from an analytic perspective, from an econometrics perspective. I can get TIF's preference for knowledge and quality of life and tease that out and compare it to Dean's preference for knowledge and quality of life. And when we think about providing healthcare according to preference, we might think of, well, what's the value of incorporating that and in terms of addressing heterogeneity? And if are there some patients who highly value it, and there's some patients who don't highly value it? And that's a large area or an emerging area of research, and we can do that. But there's, again, some ways to go in terms of really having a validity conversation around, is the science there in terms of preference research to be able to meaningfully distinguish between TIF's and Dean's preferences.
And again, the uncertainty of sample size reduces to n = 1. Yeah.
It does, but I will say to that, and so this is part of my PhD research in rare diseases, idiopathic intellectual disability specifically, is what we're able to tease out is I can get individual preferences, and it does reduce to n of 1, but as it turns out, I can appeal to the distribution of all preferences and find out exactly where TIFF is in that distribution. To some degree of probability. And so the idea is that, we pick out you within a distribution and we use the strength of the entire population to infer the value of a single individual. And so some really nice, what I'll call econometric Bayesian approaches to thinking through individualized N of 1 inference.
Very interesting. I too could talk about this all day, but I better get to questions. MJ, thank you for your question. So Dean, this is more about this infrastructural mechanisms of healthcare that can help collect these data that's going to be required. So she asks, has the federated structure of Canada impacted the plans for collecting real world evidence across its healthcare system?
Yeah, and I think Rob touched on this in August, and it's exactly the right question you need to ask. And so the short answer is, because Canada is not a healthcare system, we are a collection of healthcare systems. So each province makes its own decision and collects its own data. And there are initiatives at Genome Canada and other initiatives, for example, the Terry Fox Marathon of Hope Cancer Centers Network, that are trying to enable data sharing across Canada, across our healthcare systems, because we are so siloed. And so these initiatives are funded, and they are funded well. And even against that funding, we're still having real challenges in building out the federated analytics needed in order to analyze data from different healthcare systems. The other thing I will note is that we do this really well with genomes right now. And so we have something called CanDig, and we're able to do that right now in terms of omics and sharing omics across our systems in Canada. The quality of life data, the length of life data, the health services resource utilization data that the Ministry of Health collects are not quite consented in the same way, in terms of a clinical trial, in terms of an omics-led clinical trial. And so what we are working out, beyond the infrastructure, is just those consent and privacy components of what it means to share the entire core data set with each other in a way that is respectful of legislation, in a way that meets privacy expectations of the different provinces and the people who reside.
Well, that should be easy, surely.
We've talked about it for so long. Open science is such an important to us, and it's crazy.
No, it's a painful reality here too, as you know, and that sort of touches on the comment by Anonymous about, currently our data collection mechanisms are woefully short of facilitating this, let alone the regulatory and jurisdictional overlay there as well.
Yep. And it's just like a lot of investment is needed, and we've moved towards this investment, and we have data architects and engineers to help make this happen in BC, but it's a big investment, and it's really complicated. And my data architect, she pulls her hair out every day when I see her, saying, "This is impossible." We are trying to move forward.
Yeah. There is another question from David Moad. Whilst this is this iterative process is underway, how do we deliver safe and effective clinical genetic services to patients and families, given the excessive demand driven by sort of the technology marketplace?
Yeah, and I think that excessive demand driven by the marketplace is the reason why we created this idea of life cycle health technology assessment. Even in absence of high quality safety and effectiveness data. And so the short answer is we need to, in a research context, start to fire up our healthcare systems to collect the right data, to consent patients, because they need to be consented to participate in research. And we need just to start collecting the data. We need to start somewhere. And we think that being mindful of where we're going in terms of equity and safety and incremental cost effectiveness, we need to be mindful we're going from the start. And so I guess the short answer is we do think life cycle iterative decision making is important, but it doesn't mitigate the need for the research realm, which is really starting to kind of fire up research in context to discovery and initial trialing of discovery in healthcare patients under the watchful eye of ethics, and of course in relationship with industry, and I think in relationship with regulatory authorities.
Mm.
Start now instead of later. Yeah.
Yes, yes. Move quickly, get the momentum behind us. Now, Dean, thank you so much again for joining us this afternoon / this morning. We are nearly at time, but it's been really fascinating to hear your thoughts and approaches to health technology assessment, and it would be excellent to continue this conversation with Canada. I know you have existing relationships with ILIAS and others, so it would be fantastic to certainly work on this collectively.
Absolutely, and that's our intention.
Wonderful. And to all, thank you very much for joining us this morning. I hope you enjoyed it as much as I. Our next DNA Dialogue seminar will be held on the 24th of November, and we'll be joined by Patrick Tan from Precise in Singapore, and he will be speaking on transforming health in Singapore through precision medicine. So thank you again for joining us, Dean. Thank you. And we will see you all next month.